I'm from Georgia Tech, and we have an IGERT there on stem cell biomanufacturing. So what I thought I would do is lead off, because I think the term manufacturing has been pretty popular lately. This is something that has become one of the top initiatives from the Obama administration to really try and stimulate a resurgence in our ability as a country and to stimulate economic development to reinvigorate our ability to make things. And I think that one of the things that he has highlighted that fits well with the theme of our IGERT is actually in the advanced manufacturing area and training the workforce for these types of skill sets where the U.S. has traditionally excelled. So even as foreign countries have taken over a lot of the general manufacturing of various components that go into our technologies of today, the U.S. is still traditionally led in the area of advanced manufacturing, where a lot of the development of these ideas and the high skill set is required. But even recently, within the last couple of years, this has now started to tail off even below, and we're into a deficit now in our advanced manufacturing, even while there has been some increase in the total manufacturing that's been shown. So and just this past week, actually, I thought that this is the third call I've had now for jobs and innovation. So this is something that is of high priority to our country. Two things, too, that I just wanted to note to set up then what I want to show is some of the research areas that are integrated within our IGERT are actually come from two of these reports that have been first from the President's Council of Advisors. This is a group of CEOs of the top companies throughout the country, as well as some of the top presidents of many of the technical universities, including Georgia Tech as one of these. And what they've highlighted is the importance of being able to coordinate the use of information, automation, computation, software, sensing, networking, and building upon what is known from the physical and the biological sciences, particularly materials being one of these. And the other is actually then from the National Science and Technology Council, so slightly different group, but also reporting to the President, that highlighted specific objectives that should go into the strategic plan for advanced manufacturing. And number two on this is important, is expand the number of workers who have the skills needed by a growing advanced manufacturing sector, make education and training system more responsive to demand for skills. So I think this fits well even generally with the goals of what IGERTs are trying to do. So when it comes to stem cell biomanufacturing, then what is this? What is the need, and what is this industry? Which it's really, I would say, a training for an industry that is in its very nascent state. It really isn't an established industry at this point. What we have is basically a tremendous amount of basic knowledge that has emerged from stem cell biology, I argue over particularly the last 30 years, but going back about 60 years since stem cells were first identified in the use of that term. And what we constantly hear about, and I talk about and others in these, is all the great things that stem cells are going to do, but there's only one pathway by which that can happen, and that is you have to take things, this raw material. So much like in the petroleum industry, crude oil is in and of itself not useful until it's processed appropriately and then can yield the types of technologies directly from that, as well as the things that we now enjoy as modern conveniences, cars, boats, planes, and et cetera. And similarly, we have these envisioned types of applications of what stem cells are going to do for us. But what we need to be able to have then is this platform that in between this process takes this raw material, processes it appropriately, and also can then deliver it in formats that can accommodate these types of applications. So what this means then is that there's I would argue, increasing need to be able to start at this interface where the biology is and be able to develop the technologies that are efficient, scalable, and robust enough to enable this type of manufacturing to occur. So what is that? Well, if you put it in a traditional sort of bioprocessing sense, there's some key elements of it. There's the raw material, I said, the stem cells. There's upstream processing, which refers to all of the manufacturing side of it. How do you amplify, produce more of the cells? How can you do this in a controlled way? How do you control the phenotype? Much of this done through the form or format of how the cells can be grown in various ways, and then all the various types of ways and scale-up types of approaches that might be used. Importantly, though, also is how do you monitor what's going on? And that's where I'd say right now probably the biggest limitation in the field. You can grow cells to great abundance, but doing it well and doing it when the cell is the product as opposed to what the cell produces is a fundamental transformation in actually all biomanufacturing. And so here you have all of these great molecular tools, but the question is can you somehow use this information or come up with ways in which you don't destroy your product or all of your product as a means of assessing what you had? And downstream from that, if you produce and amplify large numbers of cells, you come up with ways in which you can sort these appropriately, ultimately then delivering them in ways that can actually feed in or directly useful for diagnostic platforms and some of the therapeutic applications which we so commonly hear about. So what I'd like to do is show you a couple of the projects then, and kind of I've put these in this order of this, starting first with two of our trainees, the first and the second year. We're 2 years into the program. So Jen Lee and Tori Rinker from the Temanoff lab, which focuses on interfacial tissue engineering, and one of the things that they have been developing that's fairly unique is a tri-layer or more systems whereby you can control the populations of cells in laminated structures, which you can then actually separate selectively through different enzymatic treatments and actually retrieve these cells and perform various types of phenotypic assessments. So this is a much more complex type of system in which you can start to actually ask questions about how one cell type or population affects another by these biomaterials-based approaches. They've applied this for a number of cell types, but in particular for the IGERT purposes, looking at mesenchymal stem cell populations, as well as cells that could then, that they may affect the differentiation of, and cells that could affect the differentiation of those cells. Going down another layer in this then is also another of our trainees who's in her second year, Allison Douglas from Biomedical Engineering, and Tom Barker's lab. The Barker lab focuses on especially extracellular matrix engineering principles, and one of the things they're particularly interested in as well is in mechanobiology. There's been tremendous interest, as many of you know, since the papers that emerged about 6 years ago in terms of matrix stiffness, but that's only one of the physical properties that cells can encounter. And so in some of the work that Allison is looking at, they're actually looking at different other parameters that can be fine-tuned by how you manipulate elements molecularly of these environments, such as viscosity, and specifically being able to do this with the types of engineering conjugation approaches they've used for different adhesive cues. And this, again, is a project that she's studying primarily on mesenchymal stem cell phenotype, but this is a principle that could be applied to other stem cell types as well. From my own lab, two trainees, Jenna Wilson, who is actually our representative trainee attending this conference and has a poster that I'd encourage you to take a look at, along with Josh Zimmerman, have actually then been looking at microencapsulation. This is a commonly examined platform in some respects, but not really utilized successfully yet or implemented for stem cell manufacturing or bioprocessing. And what both of them are interested in, and we've done this with both pluripotent, so embryonic stem cells, as well as multipotent mesenchymal stem cell populations, successfully encapsulate these, but this is really the first step in this process. What they're then looking for is how you might be able to use encapsulation to not only control the populations, culture them at high density in new types of configurations, such as packed bed types of systems, which have yet to be attempted for stem cell culture purposes, and also then be able to look at how you might be able to use this to fine-tune paracrine delivery. It's not always possible to analyze everything experimentally, at least with some of the complexities of this. And so Doug White, one of our other trainees, who is actually also in biomedical engineering but has a strong background in computation, is actually working in Melissa Kemp's lab and has created, using the experimental system of these aggregates, a 3D computational model that can capture the dynamics of these systems. So what we're excited about with this is that by using simple rules-based algorithms, you can probe the potential type of experimental space in silico to be able to screen and identify the types of rules and systems that sometimes we can't always, that are kind of underlying the culture systems. And you can probe these theoretically before then going back and designing experiments, and in theory then being able to control the environments and the systems in such a way to get more predictable and defined outcomes. One of the other very unique things I think that one of the students working on this is Greg Holst, who works in a precisions laboratory in mechanical engineering. He's actually coming up with an approach that can greatly make the gene expression, regular PCR tools, these whole cycling process much more quick, much more efficient and quick. So by doing this, what this enables for the first time is to be able to think about how if you did sample from populations of your cells, the gene expression, you could rapidly turn that information back around and start to integrate feedback control that's not based on temperature or pH or even metabolites, some of the common methods that are used, but actually what the cells and the state of those cells. And one of the things that's key is that the state of some of these cell populations can change rather rapidly over the course of hours. So to be able to meet that need, you need something that can operate on this length or this time scale. And basically what they have it down to now is something that can operate in less than an hour, something that most people would argue is probably quick enough to make the decisions that could perhaps affect the fate of this cell population. One of the other mechanical engineering students, Tom Bongiorno from Todd Solchik's lab, actually building upon a lot of the interest, not just from the stiffness of matrices and environments, but the actual physical properties of the cells themselves. And so he's been examining using atomic force microscopy initially and characterizing the stiffness of different cell populations from different commonly used sources. So adipose tissue and bone marrow are the two most common tissues used to derive mesenchymal stem cells. Those are very different types of environments where these cells come from originally, and as a result, there's differences, vast differences in the mechanics associated with it. But instead of measure those two environments, he's actually measuring the two populations and can see that there are subtle but significant differences between these. Furthermore, what he's then looking at is that you're actually with a microfluidic based device, you can sort cells based on their stiffness. And this is again a new principle that can be used, but this is a label-free, non-destructive, high throughput approach that can operate at the same speed as flow cytometry. So this is quite interesting because now again you have a way to now sort cells and not have to separate them out, but you have more information content on them. And this is the last one. Is that basically at the end of this process you produce these, how are you going to deliver them appropriately, what are the environments for this? And so two trainees, again from Andres Garcia's lab, whose lab focuses a lot on biomaterials development and tissue engineering applications, they've actually come up with, again, PEG-based hydrogel systems that can stably introduce these cells. The biggest problem with cell transplantation currently is that you typically see 95% or greater loss within a day or sometimes even shorter. This is due to the cells dying or actually just being at a site where there's a lot of blood flow and other things just washed away. So they never are there long enough to really have much potency. So the use of hydrogel-based approaches such as this in vivo, which can stably retain the cells and control their phenotype, is vitally important for the ultimate success of such therapies. So just to summarize then, working from kind of the full end of the spectrum, we have from the early culture stages and use of biomaterial-based platforms and novel ways to really control these at a very fine precision, both spatially as well as temporally. Computational tools that are being developed in parallel to really facilitate better analysis and understanding of these complex systems of multicellular stem cell populations. Also then the development of, I think, very novel types of real-time molecular analysis tools that can be used for feedback, as well as novel ways of sorting out these populations so that you have very defined products, which then can be combined with these types of robust vehicles that are being delivered or developed for delivery of the cells at the end. So I just want to again acknowledge and thank this. This has been a tremendous whirlwind of effort in 2 years of time. We're very grateful for the support.